Hello, everybody. I hope you're doing well. So I had to look at and reflect on where we were at in our course and where we needed to be right before spring break. And I noted that we weren't going to be able to get where we needed to be unless I did at least one recording. I may have to do another, but this is a recording for a lecture that you're going to have to listen to on your own time. And then you can ask questions anytime. You can call me, of course. You can send me an email, or you can ask questions about any of the material in this lecture um, during class. Okay. So this lecture is the first lecture in Unit 2, and it's entitled Cell Structure and Function. And what we're going to be looking at are the different types of cells, right? So if we look here, you can see that there's some different types of cells, and what all these cells have in common are that they are alive, right? And so the simplest unit of life is a cell. Now, what makes them different, of course, is that nasal cells and onion cells are eukaryotic, right? And so some of you may be thinking, well, what is a eukaryote? Well, eukaryotic organism are organisms that are defined by simply having a nucleus and membrane-bounded organelles, right? The other here is a bacterium. And a lot of people, a lot of scientists, if they've never taken a microbiology course, just really never study these beautiful little creatures that I love so much, um, which are bacteria, right? Bacteria are prokaryotic, and they're defined as having no nucleus, therefore no membrane-bounded organelles, right? They do have one membrane, right? The plasma membrane, and we're going to be talking about the plasma membrane at a later date. But they're very different, right? They're so simple in design, but they're really efficient in what they do. Where the eukaryotic cells are very complicated in design, um, but they have all these structures that do specific and specialized functions for them. And so that's the two different types of cells, if you think about them. Now, if you remember when we looked at phylogeny in the first lecture that we talked about, we noted that the bacteria that are made up of what we call the classical bacteria and the archaea made up three of the domains, right? And then everything else, it was eukaryotic, or the new guys on the planet, and they made up one domain, right? Sometimes we can stain these particular cells and we can see them quite well. Bacteria are a little bit harder to see because they're so small, right? They're about 1,000 times smaller than a eukaryotic cell. Um, but we can still stain them. We can still get them. We have to use an oil immersion lens, and we talked a little bit about oil immersion lens in lab. Um, but we also have other methodologies by which we can look at these different types of cells that are really, really small. And of course, the microscope, right? This is a compound microscope, this one here. It's called a compound microscope because it has two lenses. The eyepieces are the oculars, right? They have two functions, as you know. They magnify 10 times, right? They're the first lens, right? And then they also bring the field of view to the eyes. In this case, we're interested as the magnifier, right? It has a magnification of 10 times. And then the objectives themselves that are located here, um, they also have a magnification power. This is a special type of microscope, right? It's an electron microscope. This happens to be what we call an SEM. That stands for scanning electron microscope. And with this, we can see things from 1,000 to about 10,000 times the size of what we're looking at, right? And then we can see all kinds of great things. There are other tools that if you take a micro course, right, and there are a couple of our colleagues in this classroom, right? Jennifer Green and Michelle have taken microbiology, so they know a little bit more about the bacteria. But we classify bacteria, at least the ones that we're interested in, as a from a human perspective because they cause disease. We classify them as either gram-positive or gram-negative, and we'll talk a little bit about the differences in their cell walls, but that really is what makes them gram-positive, gram-negative. We use a tool called the gram stain, and the gram stain uses a purple dye, crystal violet, and things that stain purple are said to be gram-positive, and it uses a red dye, safranin, and things that stain red are said, or pink, are said to be gram-negative, right? And so, but the tool, the gram stain is just a tool, and it allows us to appreciate the differences in the cell wall composition because they're very different, and that's what makes these cells different, right? So some cells, when you evaluate them, they can be considered dysplastic, and that's really important for us to understand. Dysplastic means not normal, right? And so sometimes you might get some results and the results say that you have dysplasia or dysplastic cells. And what that means is the cells are abnormal. And in some cases, they could be a precursor or an early sign of cancer developing in that area, right? In some cases, you've just had some injury for whatever might have happened to you, and some of these cells look a little bit different. You could have had an infectious agent that were on the surface of them and they damaged the cells, and so they look dysplastic, they look abnormal. But you have to do a little bit more investigation in order to figure out if those cells are normal or abnormal. Remember, I've always told you all that disease happens at the molecular level, right? It happens at the subcellular level, right? The molecular level. And then it affects the cells, which then affects the tissues, which then affects the organs, which then affects the organ systems. And then eventually it can affect a whole organism. So as we begin to talk about these cells and really cell theory, I kind of want to remind you of what we're looking at, right? So we're mostly interested in organisms in this area here, right? Because that's where the plant and the animal cells kind of hang out, right? And you can see they're anywhere from about 50 micrometers in size, right? Whereas some bacteria, at least the larger ones, can be around 1 micron by 2 microns, right? And so they're pretty small. They're roughly 500 times smaller, or in some cases 1,000 times smaller. And then you can see you get over here into the smaller areas where you start to look at nanometers, and you start to see things like viruses. You go even lower than that, and you get to see proteins and lipids, right? And then if you get even lower than that, you're talking about picometers now. Now, we can see some molecules, but atoms are really hard to appreciate. So most of the things that we look at are really renderings and kind of hypothetical ideas of how small these things are because they're just too small, right? And so if we think about that for a minute, then that's pretty cool because you can see an adult female, the average height is about 1 meter or so, right? And for males, it's about 1.4 meters, right? So just slightly a little bit taller in height. Right? So we're looking at this most area here because this is cellular and molecular, and this course is really designed around the eukaryotic cell, where microbiology is really kind of designed around the prokaryotic cell. I'll still talk about them because some of you will not take a microbiology course for whatever reason. I believe that anybody who wants to go into science should take a microbiology course, but that's just my opinion, right? Especially if you want to go into the molecular sciences because they're so um, they're so attracted to each other. They're so ingrained with each other. They're lockstep with each other, and a lot of the techniques you would learn in microbiology you would also use in the molecular world, right? So if we look at cell theory, there's really three tenets to cell theory. Number one, all living organisms are made up of at least one cell. Now, there can be more cells, they can be multicellular, but they have to have at least one cell, right? So think about a bacterium or protist or an amoeba. Those are all single-celled organisms, right? The cell is the smallest unit of life, and if you get it any smaller than then it's not living anymore, right? So a virus is not alive, right? A virus is biologically active, but it doesn't really meet the tenets of life, right? And so it does replicate, but it really is kind of a little bit funny about that, in that it infects a cell, it takes over the cell, and it uses the cell's machinery, it uses cell biochemistry to actually make more viruses, right? So it doesn't really replicate on its own. It has to infect a cell in order to replicate, right? And it doesn't have any metabolic processes that are associated with the virus. It does have some enzymes, but not any metabolic. So just those two right there, it gets knocked out from being alive, right? The third tenet, of course, is that all cells come from pre-existing cells. And so if we think about that for a minute, in your body right now, you have two types of cells. You have what we call diploid cells, and diploid cells are somatic cells, right? Somatic means of the body, right? And so it's spelled like this, somatic cells. And that is all the cells of the body except not the gametes. The gametes, so somatic cells are said to be 2N or diploid, right? The gametes, um, also known as the sperm and the ova, the gametes are said to be haploid, right? And so they're denoted by having half of the amount of chromosomes that are associated with a diploid cell, right? But so right now in your body, you have two types of cells, right? You have haploid cells and diploid cells. You have everything that's part of your body, all your tongue cells, your eye cells, your skin cells, your kidney cells, all those are somatic cells, right? But the cells that are produced in the testes of men and the ovaries of women are haploid, right? They're gametes, and so they're different. Okay. If we look a little bit about these particular structures, right? So I always like to show electron micrographs. These are transmission electron micrographs, TEMs, and what you get to see here is a lot. You can see a lot smaller things like inside of cells, right? So if we look at the construct of what we call a eukaryotic cell, right? And so you can see it here. It's much more complicated than the simple bacterial cell. And you can see it here, right? Here's the bacterial cell right here. And you can see there's no nucleus. The DNA, you might be able to appreciate just a little bit of it, right? But it's kind of in the cell itself. It's sometimes balled up and sometimes it's unraveled. But you really are having a hard time seeing it here because we're not really looking for it, right? We're not staining for it or looking for it. But here, with this eukaryotic cell, right? This is the outside of the cell, right? And inside you can see is the nucleus, right? And then all these other structures that are in here that are real tiny, all these little things right in here, right? These are all organelles, where bacteria don't have organelles. And then here you can see the condensate, the 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 DNA, right? Which there's a lot more of it. Bacteria typically have only one chromosome, right? Where eukaryotic cells can have multiple different amounts of chromosomes, depending on the species of organism that we're talking about. So I have this chart for you guys to take a look at, and really you can see the differences between prokaryotic and eukaryotic cells. But the biggest difference, of course, is the nucleus, where prokaryotes simply have no nucleus, but the eukaryotes do, right? And then membrane-bounded organelles, they're absent in bacteria, but there's a whole bunch of them in a eukaryotic cell. And there's a whole bunch of other things you can take a look at. We're going to be talking about some of these things. But if you can remember, these two right here, right? The nucleus and the membrane enclosed organelles or membrane bounded organelles, that's really a good thing to know, right? If we look at a Venn diagram of those, you can see that there's very big differences between prokaryotes and eukaryotes, but there's also some things that are shared by them, right? And so you can see here, both types of cells have at least one chromosome. They have DNA as genetic material. They do have ribosomes. They have cytoplasm, also known as the intracellular fluid of a cytosol. They have a plasma membrane, which really kind of sets the boundaries of what's the inside of the cell and what's the outside of the cell. Uh, they can have cell walls, right? And we say sometimes because not all eukaryotic organisms have cell walls. Plants do, algae do, fungi do, right? But animals do not. And some protists do, but some do not. Where bacteria, most of them have cell walls, but there are some bacterial cells that do not have cell walls. So these are the traits that the Venn diagram here is showing that they share, right? They share these traits, but then there's quite a bit of differences between the two. And I'll take it so that you can look at these on your own time and see the differences and really appreciate the differences between prokaryotic and eukaryotic cells. Right? Let's talk a little bit about prokaryotes before we go on and talk about eukaryotes, right? So with prokaryotes, no nucleus, they have no membrane-bounded organelles. They do have characteristics that all cells have. They have a plasma membrane. A lot of them have cell walls, and some of them have a capsule. A capsule is a outer covering of some bacteria that protects them from being phagocytized or being injured. But it also, because the capsules are made out of sugars, they can be a reserve for nutrients, right? And they do have a plasma membrane, and the construction or the way that the plasma membrane is built in a prokaryote is very similar to the eukaryote. There are some differences, right? There's no cholesterol in a prokaryotic cell, and so that's one big difference. And so there are a few others, but that's really the biggest differences of them all. Also, in prokaryotes, they have PILI, and PILI are kind of used a little bit for the movement, but really they're mostly used to move what we call plasmids, right, from one cell to another. So plasmids are small pieces of DNA that are self-replicating, and they usually code for a specific metabolite, right? And especially the gram-negative cells, those organisms that are gram-negative, can extend their, I'll show you a micrograph of it in a minute, can extend their PILI, and they can share plasmids, right? And then they can share resistance to antibiotics, things like that, right? Bacteria, prokaryotes, also have ribosomes, but the ribosomes are free floating in the cytoplasm, where in eukaryotes they're attached to the rough endoplasmic reticulum, right? And so there's a big difference there also. They have cytosol or cytoplasm or intracellular fluid. All those things mean the same thing, right? And this is the fluid matrix of the inside of the cell. And they have DNA, right? But their DNA is small, right? It's usually circular, right? It's sometimes called a DNA loop. It's located typically in the center, but it could be anywhere else. It's called a nucleoid. Oid at the end of anything means resembles. And so this resembles a nucleus, but not a nucleus. And it's much smaller. The DNA material, the chromosome, is much smaller than we would find in eukaryotic cells. Right? I told you I would show you some of these structures, right? And so you can see here. Here you can see these are fimbriae, and these fimbriae are used for attachment of bacteria to other cell surfaces, right? But here you can see this is a PILI, a pilus, right? And these two organisms, right? This is a donor, and this would be a recipient, and they're sharing plasmids, right? And so let's say that particular plasmid codes for the resistance to penicillin, right? So it's resistance to penicillin, and one of them shares it with the other. Now both of them can be resistant to penicillin. Right? It's pretty interesting. It's a cool mechanism, right? And it really helps for the bacteria to be able to survive in a competitive environment. Here you can see are the fimbriae that are highlighted by this particular staining method, and then you can see some flagella, right? There is a pilus here. You can see it here. It's a little bit different than the other structures. So it's really interesting. These are micrographs, right? This is actually a phase contrast micrograph, and here is a little bit stronger microscope with a little different technique called a negative stain so that we can see the structure of the bacteria. Right. So I do have a chart here for you guys to take a look at to tell the difference between FIMBRIAE and PILI. Take a look at it on your own time. I may ask a couple of questions of these on the exam, and if you have any questions, you can ask during our lecture time together, or you can call me, or you can send me an email. Right. So just as a recap, real quick, before we leave prokaryotic cells, right? Because I know and I think and I believe that any science person should know what a prokaryotic cell is, right? They should know what a bacterium is. It's just so crazy that people call viruses bacteria and bacteria viruses and everything germs, and it just really drives me crazy. But I want people who take my courses to understand that there's a lot of living organisms out there that are much smaller than our eukaryotic cells, right? And these are prokaryotic cells, the bacteria. So the rest of this particular lecture is going to be centered around the eukaryotic cells because that's really what this course is about. And so if we think about this, let's introduce you to the eukaryotes, right? Much more complicated, right? They can be 100 to 1,000 times bigger than a prokaryote, depending on what eukaryotic cell you're looking at versus what bacterial cell you're looking at, right? They do have a plasma membrane, right? Plants and fungi and algae do have cell walls. They all have ribosomes, right? And ribosomes, of course, have a site of protein synthesis, but unlike the bacteria where the ribosomes are free floating in the cytoplasm, the ribosomes for eukaryotic cells are incorporated or attached to the rough endoplasmic reticulum, which is part of the endomembrane system of the eukaryotic cells. And we're going to talk about those in just a little bit. Right? Eukaryotic cells have cytoplasm, cytosol, intercellular fluid, right? They have DNA. They're linear and they're arranged in chromosomes, right? And so during what we call mitosis, because unlike bacterial cells that don't have a cell undergo binary fission to replicate, eukaryotes undergo mitosis, right, for all somatic cells, and meiosis for those things that are gametes, right? So there's different mechanisms here. And we'll be talking about those different mechanisms in Unit 3, right? And then eukaryotic cells, just as a real easy way, are usually much larger than prokaryotic cells. Some of the more common kingdoms of eukaryotic cells are the protozoans, right? The fungi, the plants, and the animals, right? And in here, there's a bunch of other things, all the organisms you would have cared about, right? So all those beautiful little fur babies, the kitties and the dogs and things like that. And um, if you're Helen, you're gonna love reptiles, right? And so they would all fall into these categories here, right? And so they're really interesting to think about. So the eukaryotic cells are much more complicated. So here we're seeing plant cells and we're looking at animal cells. One of the differences right away, of course, is that plant cells have a cell wall, but animal cells do not. And so if we think about that for a minute, then if animal cells only have membranes, and for like bacteria have bacterial cells too, so we can use antibiotics that are going to attack the cell wall. And the most common or the most famous of all antibiotics is penicillin, and it attacks the cell wall, right? The other thing you might want to see here is that we have these vacuoles, right? And in plant cells, and this is a central vacuole. There's a difference between the two cells where a lot of times the animal cells are much smaller than the plant cells. So the cells have to deal with waste, and so because the plant cells are larger than animal cells, their organelles are closer to the plasma membrane, so it's easy for them to get rid of waste, right? Where the plant cells are much larger, and so you have to have these central vacuoles that just kind of push all the organelles closer to the plasma membrane. and therefore they can get rid of waste, right? Because if they don't do that, then the cells are going to die. And so that problem we like to refer to as that problem with surface area to volume ratio of cells, right? And so remember, the animal cells are smaller, so they're easier to get rid of waste. The plant cells are larger, harder to get rid of waste, and so they have a central vacuole so that they can deal with the waste problem. So, because remember, every time a cell undergoes cellular respiration, they're going to produce end products of metabolism that are not good for the cell. And if they don't get rid of those, then the cell becomes polluted and they die. And so this is one of the ways that these cells deal with it, right? So again, animal cells much smaller, so their organelles are closer to the plasma membrane, which allows them to get rid of waste. But plant cells are much larger, and therefore um, they have to deal with getting rid of that waste. And so that's an easy way to talk about how these cells might get rid of waste, right? Okay. And so as we continue on to think about how that might happen, right, the bigger cells have to have some mechanism that's going to push the waste from inside the cell to closer to the plasma membrane, and therefore they can get rid of it, right? And so we call that your surface to volume ratio, right? It's a problem for plant cells, not necessarily for animal cells, but for plant cells. And so therefore we have these different mechanisms that these cells have evolved. And that's really cool to think about how these cells have evolved to have these different mechanisms, right? Animal cells, there's a lot of them, but they're smaller and they can deal with waste, right? And if you think about how even we as multi organ system organisms deal with it, right? So the waste gets liberated from our tissues and from our blood. Our blood picks it up, takes it to the lungs, gets rid of it that way, but our blood also goes through the kidneys where it gets picked up there and it gets made into urine and we get rid of it that way. And that doesn't really talk about the digestion process in humans, but here's something might blow your mind right here, is that the alimentary canal, the digestive tract of humans is not really on the inside of the body, right? You have this opening the mouth and then you have this opening the anus, and that whole hollow tube never makes contact with the inside of the body because there's a lot of really toxic stuff and a lot of bacteria that are in there that if it were to get into the bloodstream, we would be in serious trouble, right? So that's a whole different science, right? That's physiology and anatomy. And so some of you guys may go that route if you decide to go to medical school, or if you decide to become a PA, or, um, individuals who are going to be nurses or nurse practitioners, or, and, all kinds of different things, right? And so it's really cool, right, to think about how these things work. Remember, animal cells are small, and so they can get rid of waste pretty easily. But plant cells, they have to have a central vacuole that pushes everything closer to the plasma membrane, and therefore waste can come out of the body, right? And so if we think about how that might be, right, this is really cool stuff when we think about all this stuff. Let's talk a little bit now about some of the other structures of the cell, and we're going to look at the plasma membrane now. This is the plasma membrane eukaryote, and the reason we know it's a eukaryote is because of this right here, cholesterol. Cholesterol is only found in a eukaryotic cell that's an animal cell. It's not found in plant cells, and it's not found in bacteria, right? So that's a quick way to differentiate it from all other plasma membranes. Now, if you look at these, you see that there's a lot of different proteins in here. We're going to be looking at some of those proteins, right? And we're going to be talking about how those proteins function, right? Some of them are stabilizing proteins, some of them are channels, some of them are receptor proteins. There's all kinds of different proteins in here, and we're going to be talking about those later. But really, the plasma membrane is designed because it's really a phospholipid bilayer. So if you follow my line here, I am drawing a line through really the plasma membrane, and I'm differentiating the two leaflets, the two parts of the plasma membrane, right? So this would be part one, and this would be part two, if you will. And they have these phosphate heads, and the phosphate heads are said to be hydrophilic. They like to come in contact with water, right? And so therefore water comes in contact with them all the time. Although water does not go through the membrane because it's polar, and the middle portion of the membrane, because they're composed of phospholipids, right? It's a phospholipid bilayer. So the middle portion of it is said to be hydrophobic because it doesn't like to come in contact with water. As a matter of fact, water won't get near it, right? Therefore, water has to go through a pore, an aquaporin, in order to get there. And we'll talk about that a little bit later in the semester. But this is a very beautifully designed thing. It's said to be semi-permeable. And what that means is it's going to allow certain things to come into the cell from the outside of the cell. This is the outside of the cell. This is the inside of the cell, right? Certain things can come in through it, right? And those are usually small molecules that are uncharged and that are hydrophobic or lipophilic, right? Those things can come through, like cholesterol, right? And so other things are not going to be able to come through. So any charged things like your electrolytes are not going to come through. Water's not going to come through, and large molecules like glucose or amino acids or things like that, they're not going to come through unless they come through a pore or a channel, okay, or a carrier. Okay, we'll talk about that later on in the semester. So real quickly, you can see kind of the construct of a eukaryotic cell, right? And so you can see how this is put together. I don't particularly like this. I'm probably going to get rid of it, but I'll show you this one because it's a much better representation of the actual cell itself, right? And this is actually the nucleus, right? And we're showing number one, the nucleus, and you have a nuclear envelope in the middle here. You have what we call the nucleolus, and all these things play a role. And this is part of the endomembrane system. This is the endoplasmic reticulum, the rough one, and you can see these little studs throughout are the ribosomes, right? So if we then think about going from looking at kind of the way that the nucleus is arranged with the endomembrane system, right? And how this represents that, right? So you can see the nucleus, it's large, right? It's the largest organelle of a cell. It contains the DNA, right? Um, and it's necessary for the cell. It has it's bipored. It's a phospholipid bilayer. Also, we call this the nuclear membrane, right? And it contains the DNA, but it also contains proteins that makes RNA, right? Certain types of RNA and certain types of ribosomes. Right? If we go on and think about it a little bit more, the nuclear envelope itself protects the DNA from being broken down by enzymes we call DNases, right? And it's double membrane, right? It has pores in it, right? So these pores will allow things to mostly leave, right? And so the things that leave, of course, are going to be messenger RNA. And so if you don't know what that is right now, that's okay. We're going to talk about that in Unit 3, okay, when we talk about transcription and translation, right? So the nuclear envelope protects the DNA, right, from being digested by DNases, but also when there are copies of the DNA made by messenger RNA, it allows that messenger RNA to leave the nucleus and then go to the ribosomes where the proteins can be manufactured. Okay, so there are pores again that talked about, and these pores are in the membrane itself, right? And then the DNA can't leave, but there can be movement in and out of the nucleus, right? Um, with things coming in, but also things leaving. Right? It's pretty interesting to think about. Therefore, it's a membrane-bounded organelle. The nucleolus itself is right here, and the nucleolus is kind of this darker structure, right? It sometimes you can have a double nucleolus, right? In some cases, right? It doesn't really have a membrane around it. It's protected also by the nuclear membrane, and it's involved in the process of RNA and making ribosomes. Okay, so this is a TEM showing the structure of the nucleus and the part of the um, endoplasmic reticulum, part of the endomembrane system, but you can see how all these things are constructed here and how beautiful this particular uh, micrograph is, right? It's really, really nice, right? And then here you can see what we haven't talked about yet is a Golgi apparatus, but here's just the micrograph showing it. Which brings us to talk about the endomembrane system, right? The endomembrane system consists of the endoplasmic reticulum, right? The nucleus and the endoplasmic reticulum, both the rough and the smooth, and then it's also the Golgi apparatus, right? So if we think about how all this might occur, right? You have messenger RNA being made here, right? And then they leave through a pore and they come out and they join up with one of these ribosomes, and then you have proteins being made, right? Through in the rough endoplasmic reticulum, right? And then these proteins can go into the cis face of the Golgi apparatus, where they can be modified by enzymes that are in the Golgi apparatus. And then eventually, as they go through this membrane system, right, eventually they can come out as a vacuole on the other end. And these vacuoles can go either to parts of the cell that need the proteins, or if they're just don't get, if they're not needed, then they can actually fuse with the plasma membrane and then can be liberated as waste and come out of the cell, right? So there's a portion here that's you're not being able to see, which is the smooth endoplasmic reticulum. I'm going to talk about that in just a little bit, right? So if we think about this, right, the endomembrane system consists of the smooth endoplasmic reticulum, the rough endoplasmic reticulum, the Golgi apparatus, and the transport vessels, right? But it all starts, of course, with the nuclear envelope. All these membrane systems are here, and each of these structures have a different function within the cell itself, right? So if we look at the endoplasmic reticulum, right, endo means inside, plasma means inside the plasma membrane, reticulum means a little net, if you will. So this endoplasmic reticulum is found inside the cell, and there are two types, right? And so it's beautiful to think about how this works, right? So I just kind of drew this. This is a better representation because my little arrows just go crazy sometimes, but you can see how this mechanism works, right? So you have the messenger RNA coming in, becoming associated with a ribosome, proteins being made, and then the proteins go into a vessel, and they can be modified by the Golgi apparatus. And then once they leave the Golgi apparatus, they can either be sent to parts of a cell that need these proteins, or they can be liberated to the outside of a cell because they're not needed by the cell. Okay, so rough endoplasmic reticulum, right, contains ribosomes, sites of protein synthesis. It's contiguous right up close to the nuclear envelope, which also has a little bit of ribosomes associated with it, right? And therefore proteins are made here. And if we look then here, you can see that this is the ribosomes that are on the endoplasmic reticulum. This is your messenger RNA. It's being read, and you're making a protein. These are a series of amino acids that are put together in a chain, and eventually they fold. If you recall, when we talked about proteins, you have your primary structure all the way to your quaternary structure, right? Because they're the more complex proteins. And so all this starts at the ribosome. Rough endoplasmic reticulum associated with ribosomes, and they're responsible for making proteins. Smooth endoplasmic reticulum, right? It has enzymes for metabolic processes, and it can synthesize lipids such as phospholipids, which are used to make membranes, right? In ovaries and testes, they can make certain types of hormones, right? In liver, they can detoxify, right? They can remove alcohols or poisons from the body, right? And also they can store calcium ions in muscle for the calcium potassium pumps, right? That are necessary for the muscles and other things to work properly. Right? So we talked about the nucleus and the nucleolus. We've talked about the rough endoplasmic reticulum and the ribosome. We've talked about the smooth endoplasmic reticulum. And so as we go on, we're going to talk about the Golgi apparatus, which is a series of flattened membranes, right? And what some people call it kind of the receiving or the shipping area of the cell, because as these different proteins come into it, they can be modified, right? And they can be made into whatever or changed however they need to be changed, and then eventually they get shipped off. These different proteins can be shipped off in these other vesicles to the cell, or they can be removed from the actual cell itself as a waste, right? And so you have these transport vessels, and so you can see here from the nucleus coming into the endoplasmic reticulum rough, coming associated, making proteins. The proteins are then pinched off as a vessel, or okay, then they go into the cis face of the Golgi apparatus. They get modified, they come out, and then they can go to other parts of the cell, or they can be liberated as a waste into the extracellular fluid of the cell. Okay, so I kind of have this. I've talked about it already. I've got it in steps for you guys to take a look at it, and you guys can take a look at this at your own time. But I did want to show you kind of the ribosomes themselves, right? They're going to be where your messenger RNA comes in, associates itself with it, and then you have proteins being made because different amino acids are joined, and eventually you have these proteins, right? So the ribosomes are found associated with the rough endoplasmic reticulum. Um, we can also see them free floating, but typically they're associated with the rough endoplasmic reticulum. Okay. Here's a little video for you guys to watch the endomembrane system at work. And so if you want to, you can, this particular presentation is located on Blackboard for you. You can take it, you can copy that and put it into your browser, and then you can watch this particular video. Some of the other vesicles that we see in here are lysosomes. Lysosomes are really structures that can house different things, right? And they can house either proteins or other structures, and they have digestion enzymes in them, and so they can either destroy things or they can modify things a little bit. Usually, some of these things are involved with the process of phagocytosis, especially in an animal like us, right? And so when they're involved with those, then they can destroy different bacteria or other things, right? When they come into the cell, they can destroy them and get rid of them, right? But there are some different diseases that are related to lysosomes not working properly, right? And so that's Tay-Sachs disease. And I don't know if you've ever seen an individual with Tay-Sachs. They don't usually live very long, right? They're infants, and then they get diagnosed with Tay-Sachs, and then they really struggle for the short period of time that they're on this earth. If you want, you can look up Tay-Sachs disease on YouTube, and you can check it out. Or if you want to, you can let me know, and I can send you one of the videos I typically use for other courses to talk about Tay-Sachs. But it's really about some of the lysosomes not working properly. I talked about phagocytosis a little bit here, and so you can see here. So here's a microbe, right? Here's a phagocyte. This is the phagocyte right here. And the phagocyte is going to be attracted to this microbe because it has different surface proteins on its surface where it's considered to be non-self, right? An antigen, if you will. It's easier to remember it as a non-self. And so because it's non-self, then the immune system, the phagocytes, know that it's not part of it. And so it'll bring it into the cell, right? Because it's attracted to it. It's chemotaxic. It's attracted to it because it's different. It brings it into a cell as a phagosome. So it comes in as a vesicle. And that phagosome kind of fuses with a lysosome. Remember, lysosomes have those digestive enzymes with them. So you have now a phagozyme kind of forming a union with the lysosome. So you have a phagolysosome, right? And the phagolysosome then has these digestive enzymes. It breaks down the microbe, destroys them completely. And then when they get destroyed, whatever's left over residually from those microbes, that phagolysosome will fuse with the plasma membrane of the phagocyte of that macrophage, and then all of that waste is removed into the extracellular fluid of the cell. And so for our case, this makes a lot of sense if we've had an infection or we've had some tissue damage, right? You work out a lot, you run a lot, or you were in an accident and had a lot of tissue damage like I did. I fell on the ice the other day and I have this big old bruise on my left side, right? So if we were to look at my urine, we would see a little more protein in it because all that all that all that damaged tissue is being replaced by my body right now, and the circulatory system is getting rid of that. Now, can't get rid of that through the lungs, right? Because that's just a gas release. So he gets rid of that through the kidneys, and sometimes a little bit is happening in the liver, but mostly the kidneys. And some of that protein is being liberated into my body, and that's an indication. Usually the kidneys are going to hold back protein, but when there's that much, some of it gets into the urine, and that's an indication that that there's been some damage to the body. That's a pretty cool thing. If you understand science and the way it works, then you can use these different techniques to understand how the cell is being affected, or how the cells are being affected, or how the tissues are being affected, and how the body is repairing your body, your cells. Okay. So from here, we go into talking about the mitochondria, right? So this is an electron micrograph of the mitochondria showing the outer membrane, the inner membrane, the cristae, the folds, and the matrix, right? And so we're going to be talking about this when we talk about cellular respiration a little bit later. But if we look at that a little closer, you can see the outer membrane of the mitochondria. You can see the inner membrane, right? The cristae are the folds, right? And then the matrix is that fluid area within that particular organelle, right? And this in eukaryotic cells is the place where most of the energy in the form of ATP is recreated, right? So we have ADP that's been broken apart, right? It was ATP, got broken apart, became ADP. It was adenosine triphosphate, it got broken apart. That high energy bond got broken apart, that phosphate got broken apart. Now it's ADP, adenosine diphosphate, and a phosphate. And the cells can do work. But now that molecule needs to be put back together, and it's the job of the mitochondria to do so. And so it's a really great job to have. What was really cool is that the mitochondria have their own DNA. It has one circular chromosome, right? And so that's pretty interesting to think about. And I'm going to bring this back up a little bit later as we finish today's lecture. We'll talk about that again, right? So if we think about how this works, right? So you have your outer membrane, right? Surrounding the mitochondria. We have the inner membrane, right? And the folds are called the cristae. The matrix is that fluid-filled center. And this is all part of the organelle that recreates or generates, if you will, ATP energy. Okay. Chloroplasts are important also. And so some people are confused when they say, well, chloroplast is how plants derive their energy. That's not true. Chloroplast is the way that plants derive their glucose, their sugars. They still have to go under cellular respiration to make their ATP, right? So it's a different mechanism. And really, to me, we're going to talk about photosynthesis in this unit. It's really a very inefficient process, but it's really fun to talk about, and I think you'll enjoy it when we talk about photosynthesis. So that's coming up. This will be found on anything that's photosynthetic, that's eukaryotic, right? Chloroplasts are not found in prokaryotic cells. Some prokaryotes, some bacteria are photosynthetic, but they have bacterial chlorophyll. It's a little bit different, right? It's a pigment, and so it works a little bit differently. If we look at that extracellular matrix, right, you can see it's got all these types of support tissues and fibers associated with it itself, right? And it has also on here proteins and carbohydrates, and that's how our immune system knows what belongs to us and what doesn't belong to us. Now, part of that fibrin network, part of that is the cytoskeleton, right? And so these are kind of networks of protein fibers, and they're organized uh, around the cell, right? And sometimes right on the inside of that right on the inside of that cell. And they really act to support the cell itself. And so if we think about the function of the cytoskeleton, right, it maintains the shape of the cell, right? It allows the movement of a cell, right? It's the roadway for transport vessels, right? So they can follow them, attach to them, and then go from one part of the cell to another. And then they can anchor and stabilize the nucleus, right? And other organelles so that they kind of remain fixated within certain parts of the cell. Right. And so if we were to take a look at that, you could see how it's all structured, right? So here's the plasma membrane, right? And then you have the endoplasmic reticulum, right? And then you have all this support stuff, which really make up the cytoskeleton. They're made up of microtubules, microfilaments, and intermediate filaments, right? And so if you were to take a look at these things and just look at nothing but those particular filaments and how they're protecting the cell, it'd be really, really interesting to see them, right? So you can see the intermediate filaments and how they're arranged, and the microfilaments and the microtubules, right? So microfilaments are thickened, right? The core, the cortex around the inner edge of a cell, and they're kind of like rubber bands. They resist tension, right? The microtubules themselves, if you think about them, and they're found interior to the cell, and they maintain the shape of the cell, resisting compression forces, right? And then the intermediate filaments are found throughout the cell, and they hold organelles in place. So that's their function, right? And so if you think about them, then the microfilaments are composed of actin, right? And these actin subunits are really what are the support mechanisms for the for the microfilaments. Okay, and so you can see how even these components, these actin things, can really help to do all these different things for different types of cells, right? Maintain the shape of the cell, used in muscle contractions for multicellular, multi-tissued organisms. They allow for cytoplasmic streaming, right? And hopefully we'll get to see some of that. They form pseudopoda, right, which allows for dendritic cells in the body, but also macrophages and phagocytes, but also amoeba, right? And they also form cleavage furrows as the cells dividing during mitosis, and we'll talk about that a little bit later. The microtubules themselves are the largest of the cytoskeleton components. They're made up of tubulin, right? They are made up of a protein, right? And they also function and maintain a cell shape. They're used in cilia and flagella to support them. They make up spindle fibers for mitosis, right? And so they help the mitotic process, and they help organelles and vessels move around. Um, and if we think about that for eukaryotic cells, right, you can see that here we have some flagella, right? Here's an electron micrograph of that organism. Excuse me, I'm talking too much and my throat is getting a little dry. But then we have the ciliated fibers or structures of things like a paramecium, right? And so if we think about how these microtubules are arranged and how they support them, they form kind of a 9 and 2 array, or a 9+2 array of microtubules. And so if you were to count these things, right, you could see that there's 1, 2, 3, 4, 5, 6, 7, 8, 9 pairs plus 1 pair in the middle. And that's what gives the eukaryotic flagella and cilia all this support, where these are not found in bacteria at all. And so these things are a lot stronger than you would find in a bacterial cell, right? And it's really kind of interesting to think about how that might work. Not only that, but then you have tubulin, right, which is that protein, right? And so you can see these polymers of these tubulin, which also support which also support the flagella and the cilia, right? And so if we think about intermediate filaments, right, they're made out of different proteins and they maintain the shape of the cell and they also anchor the nucleus, right? And so you can see how they're constructed, right? And they're pretty interesting to think about. Now, if we think about vacuoles, right? So there are different types of vacuoles. There are food vacuoles that eukaryotic organisms have. Some of these are food vacuoles and they're storage vacuoles for nutrients, right? They're contractile vacuoles. And some of the aquatic organisms like paramecium and some other organisms have these contractile vacuoles that allow them to deal with the problem of osmosis, right? You have all this water coming into the cell and the cell has to get rid of them. So they have these contractile vacuoles that they just simply all at once will pump all this water out of the cell so that the cell doesn't really have too much water in it and therefore burst, right? And so it's pretty cool to think about. And then we have these central vacuoles in plants, and the central vacuoles are really large. And their job, remember, is to move everything that's inside the cell closer to the plasma membrane, and therefore there can be exchange of nutrients coming in, but mostly waste coming out. And so here you can see a microscope image of the chloroplasts and the cytoplasm and the cell wall of plant cells. What you can't see is a central vacuole, right? But it's there and it's pushing everything closer to the edge of the cell, closer to the plasma membrane, so that it can be exchange of those particular waste and nutrients coming in. So as we get close to the end of today's lecture, right, we do want to mention that there are certain things that are found in the intestinal tract that are called microvilli, and the microvilli are part of the surface of the small intestines, and what they are important for is to take in nutrients, which then can be kind of put into vessels into the bloodstream, and then the body can use those particular nutrients, right? So the microvilli are the absorptive, right? So if you think about our intestinal tract, right, let's mouth, you consume things, you ingest stuff, right? Digestion happens, but only a little bit, and with carbohydrates only. Goes down the esophagus, gets to the stomach. The stomach acids then start to really break down some of the structures. But the true digestion process happens in the small intestines. In the small intestines, you have nutrients being transferred from whatever you're consuming as a nutrient, gets into the bloodstream, and then the bolus of whatever's left over kind of travels through the entire length of the small intestines, and eventually all the nutrients that can be derived from it are taken and put into the bloodstream, and then it goes into the large intestines. And in the large intestines, the function of the large intestines is to reabsorb water, right? So that water's not really lost, right? And then what you have left over, of course, is feces, and feces is waste. And so that's one way that the body gets rid of waste. And so it's a beautiful mechanism to think about. So if we think about DNA, and why there can be, 6 miles of DNA in every single cell, and why it all fits in the nucleus. And that's because we can have this DNA kind of wrap around certain types of proteins that we call histones. Histones are only found in eukaryotic cells, not in prokaryotic cells. So these histones, you can have this DNA wrap around it, and so therefore it can condense, right, and form these real tight packages. And so the DNA then gets to fit into the nucleus, right? And you really never really appreciate that there's 6 miles of DNA in every one of our cells. That's just crazy to think about how there can be that much DNA, right? There's centrioles, right? So the centrioles really attach to part of the DNA molecule when the DNA is being divided. And the centrosomes then consist of 2 centrioles, right, at right angles. And each of these things really are supportive and allow for DNA to be pulled to different poles within the cell so that it can be replicated and then, of course, divided or put into 2 different cells, right? And so as we begin to finish today's lecture, I want to talk about a little bit of intracellular junctions, and we're going to talk about a little bit about plasmodesma. We're going to talk a little bit about about gap junctions and tight junctions, right? And then also desmosomes. So the plasmodesma, that's plural, that's more than one. Plasmodesmata is singular. But you see these are typically found in adjacent plant cells, and it allows for different molecules or different nutrients to go from one cell to another, right? And so that can happen because of these junction between these two different plant cells, right? There are tight junctions, right? And the tight junctions are found adjacent in animal cells, right? They create these real tight junctions um, for adherence, right? And so you can see that when these things are like this, then you can have these found typically in skin's epithelial cells or other types of cells like that, right? And so then you can have um, these tight junctions stop different things from happening. For instance, tight junctions on the epithelial lining of the urinary bladder will prevent urine from leaking into the extracellular space, right? And so that's a pretty interesting mechanism to think about. In desmosomes, right? So here you have these desmosomes that are typically found in animal cells, right? And really think about them as they're just kind of a weld between two different epithelial cells, right? And so as we think about this, they're created really by cadherins, and cadherins just mean calcium that is actually being used to adhere to the different portions of these two different layers of the desmosome itself, right? So the cadherins, it's got calcium in it. That's why it's called Ca for calcium, and then adherents is kind of a cool thing to think about, right? But these help keep cells together and kind of a sheet-like formation in organs and tissues that stretch, like skin and heart and our muscles, right? And so they're really great to have. So therefore, if you lack calcium and sodium, you can develop muscle cramps because the adhering mechanisms of the muscles aren't working very well, right? And lastly, another junction is called a gap junction, right? And so these are similar, right, to plasmodesmata and plant cells, right? But these are in animal cells, right? And they allow transports of nutrients and ions, right, uh, from different cells, right? And so they're called gap junctions because they're found and forming a gap between two different cells. Okay. The last thing we'll talk about today is, remember I told you that the um, mitochondria have their own DNA, and that's because mitochondria and chloroplasts, we believe, were once bacterial cells, right? So this is called endosymbiotic theory. And so endosymbiotic theory basically states that a whole bunch of long time ago, when you had all these prokaryotic cells, because if you remember phylogenetically, when we looked at that phylogenetic tree, it appears that eukaryotes came from the archaea, right? And so what we believe is um, that we had these archaeocells forming a relationship with each other, right? The symbiotic, right? And so some of these cells got into other cells, and when they got into other cells over a period of time, long time, right, they stopped acting like cells and started doing specialized functions for cells, right? So in some cells, the mitochondria just stopped being a cell, and they became the place in the eukaryotic cells where DNA was created, right? And and in plant cells, right, and algae, um, the chloroplasts, which we also believe were bacteria at one time, stopped being stopped being a bacterial cell and started doing a specialized function. In the case of plant cells or algae, they became the place where glucose was made, right? They became the photosynthetic element of the cells. And so that's really interesting to think about how that might have occurred, right? And so this is kind of showing how that might have happened, right? But we know that this is really interesting for a couple of reasons, right? Is that if you look at the structure of a mitochondria or the structure of a chloroplast, they're structurally very similar to a bacteria. And the other thing to think about, just think about mitochondria, is that they have their own DNA. They have one chromosome, just like a bacterial cell, right? What's really interesting to me is that um, you can look at the mitochondrial DNA and figure out who your mother is, because we as animals only get mitochondrial DNA. We only get mitochondria, really, the mitochondrial DNA, from our maternal side of um, of the relationship, right? That forms us. And so it's really an interesting thing to think about what I just brought up, right? And so you can see that we believe that these symbiotic relations led to the formation of these organelles, the mitochondria, and also the chloroplast. You can take a look at this, pull this in there, and see if you can visualize the video that will show you how we think endosymbiotic um, relationships occurred. Um, and let me know if that particular site's working, because it may not be working anymore, right? So when we think about endosymbiotic theory and evidence that supports it, right? So there's both the mitochondria and chloroplasts are similar to bacteria in size and structure. They're bounded by a double membrane. They contain a limited amount of DNA, one chromosome. They divide by splitting. They have their own ribosomes, right? They produce some proteins, and ribosomal RNA sequence is similar in prokaryotic ribosomal sequences, right? And so all that leads us to believe that the mitochondria and the chloroplasts were once bacteria, but are now specialized organelles in our cells, in eukaryotic cells, right? I don't all mitochondrial DNA is inherited from a mother, right? So, um, and then I found this interesting tidbit, right? A while back, mitochondrial DNA analysis showed that 14% of DNA in our cells we had in common with Neanderthals, right? So remember, um, organisms that are related to each other share lots of different things, and so we share DNA with our ancestors that we once called um, Homo neanderthalis, right, which is the Neanderthal, and that's pretty interesting to think about. Although some people don't want to be called a Neanderthal, but really you shouldn't take that too seriously if somebody calls you a Neanderthal, because we all have a little bit of Neanderthal DNA in us. Right. Last slide is a slide that really compares prokaryotic and eukaryotic cells. Take a look at that when you have time. And if you have any questions as you're watching this video, you can be sure to call me, email me, or ask me when we meet back in lecture, and I'll be happy to try to address those concerns for you. Okay. This is the end of the material for cell structure and function. We covered eukaryotic and prokaryotic cells, and some of the more important structures within them. We didn't cover everything, but we covered some really good stuff, right? And so again, if you have questions, let me know. But this is the end of the material for this particular lecture, right? Y'all stay safe. Pro V out. Bye-bye.